I don't really do much social media.
But you know what? You would be good at it. And I can't tell you how many beekeepers are on social media. It's just, especially Instagram, that you get to see pictures of everything that they're doing. It's really
It's a good place for them there. They do no harm out there.
All right, so we are live on YouTube as well as Zoom.
Awesome.
Oh, wow.
Is it 6:30 officially? Nope, got 5 more minutes.
So you can log into YouTube to watch a Zoom meeting?
This is just so much easier than trying to do it in the room and having to deal with 4 million cables.
Yeah. Well, we appreciate your technology efforts, Paul.
Frankly,
Absolutely.
what would we do without Paul?
That's right.
You like my little logo up here?
Yeah,
I
very
do.
nice.
I see that. Yeah. I should use that for some of my work calls.
Good evening, people.
Yay,
Hi, Jeanette.
Oh.
Are you there? There she is. Yay.
There you are. Let's see. Dave Z joined us.
Dave Z.
Hey, Dave. Good to have you.
Yes, absolutely. Hello. I'm going to be eating,
Dave.
so I'm going to put myself on mute so I can eat my pasta.
This is dinner theater. Jack and I had some eggs earlier. So.
You
Hey,
did.
if you need eggs, Jim, come get them from me. I have enough.
Oh, yeah?
Yeah.
Oh, did you
Might
get some
pick you
new
up on that.
chickens?
I got juvenile chickens from Karen right after, or right at Christmas time, and they finally started laying. So I got 9 chickens from her in the pitch dark, which is when you catch wild chickens, right? And brought them home. And of the 9, 3 are roosters.
Nice.
Just what you wanted, right?
And they're
Those are also-
beautiful. They're just beautiful. So if anybody
Also known
needs
as
a beautiful
fryers.
rooster, huh?
Also known as fryers.
Well, so anyway, I have 6 hens. I get 6 eggs a day. So I don't have a lot of eggs, but I have enough eggs. I'm excited.
Wonderful.
And if anyone needs a rooster, I have those too. And they're pretty.
And tasty.
Well, I don't know about that. They're big and they're young.
My daughter-in-law said she wanted some chickens, and I'm not really, she's in Kansas, and I'm not really clear on how it happened, but she now has over 30 chickens.
Oh, that is very common. It's chicken math, like beehive math. I want to get a hive of bees. Well, 2 hives is good. Well, 7 hives will work. Chicken math.
Yeah.
Well, that's part of the problem. If you get to be a good beekeeper and your bees don't die, then they split. I mean, you gotta have a plan for what you're gonna do with the extra bees.
And as Karen and I testified, you never have enough woodware, ever, 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 ever. Except maybe now I might.
Mm-hmm.
I have a stack in the barn. So there's Pete.
Well, but you probably end up not having what you need.
Or not enough.
Well,
Yeah, not enough of one thing that you need.
right. And there is a lifespan to it, man. Those bottom boards and the outer covers, they just don't last very long. I was thinking
I don't
about
have
going.
any problem with the outer covers, but the bottom boards are a problem.
Well, I was
Yeah,
thinking
the
of
ones-
going to
The
those.
ones I got from Kelly just fell apart.
Yeah. I noticed some of the newer outer covers I got are not as deep as the old ones. The old ones are like this deep with the metal. The new ones are like this deep with the metal.
Yeah, cutting costs.
Yeah. And of course, it doesn't sit down as far on there, and I don't like it.
David, I saw you peeking in.
What are you doing?
Tried.
Say yes again.
Yes.
Good to see you.
Yeah.
Okay.
That's a cat.
Looks like Jack's had enough. Yeah, we're changing the topic of the meeting tonight.
Yeah.
How many people's here so far?
Are you going to talk, Jack?
It'd probably make as much sense as I would.
There are 18 so far, looks like.
He's trying to decide whether he can stand on the keyboard.
I bet he can.
All right, buddy. Off you go.
6:29. We've got 16 participants so far.
I don't even know what's behind me.
Hello, everybody.
Hello, Jeff.
Hey,
How you doing?
hey, Jeff.
Kevin says hello.
Hey,
Thomas,
how's it going?
how you doing?
Good.
Hey, Thomas.
Hey,
Paul, did you do a test with our speaker tonight? Do we know that we're going to be?
I did not do a test with the speaker.
Okay. What is he? I don't remember his name. Is he logged in?
Eric.
Yeah, he's here. I just saw his name pop up.
Okay, good.
All right, you ready for me to go?
We just need, yeah, because you need to be muted for the presentation.
All right. Okay, well, you ready? Should we go?
Yes.
All right. Thanks everybody for being here at our January meeting. This is North Central Beekeepers Club. I am Tracy Pielmeyer. If you haven't met me already, I'm glad that you're all here, and just want to give a couple announcements. Next month, February, our February meeting, we do plan to have our Beekeeping 101 going on simultaneously with our regular meeting. So if you're interested or have friends who are interested in Beekeeping 101, we are hoping that will be an in-person meeting, and 101 will be going on at the very same time as our regular club meeting. Number two, Beekeepers of Indiana are having their bee school the last Saturday of February, which is the 26th. It will be up in Muncie this year. It is going to be an in-person event. They're only allowing 250 people. And at least 50 of those people have to be beginners signing up for the 101 track that they offer all day. So if you would like to attend the Indiana Bee School, you need to go to their website and get signed up as soon as possible so they know how many people are coming. I will give my annual lecture, which I will give probably every month, which is if this is your first year to do beekeeping, do not worry about getting bees. Do not be under the tyranny of urgency thinking there will be no bees and oh my gosh, I have to sign up for a package from California immediately or we won't get any. That is not true. We have lots of good local suppliers, and I can assure you that you can get bees this year still buying local, which is what we would encourage you to do. RJ Honey in Lebanon is also offering a once a month beekeeping class. It's on a Saturday each month from February through October. It is not a repeat each month. They will be going through what a beekeeper does during that month. So it's something that you would want to continue on or just catch up on the month maybe that you haven't done whatever they're working on that month. So that's RJ Honey in Lebanon. Pat Sollenberg told me she has a great list of local suppliers that she's going to share with us that we'll be posting on our Facebook page. So if you are not in our Facebook group, please go sign up. That is not the same as our original Facebook page. It is a group. You have to request to join. You have to agree and then answer 3 questions. If you don't agree and answer the 3 questions, we're not going to let you in. So that's to avoid bots. Please sign up at our Facebook group. I think that's all I have.
Okay, I guess I'm next on the agenda. Many of you probably knew Barry McNulty, or at least if you've been around the club, you'd seen him. Big guy, very jovial, very loud, very involved. Barry died on December 17th of last year as a result of COVID. You know, we as a club, we haven't really met in a forum where we could say anything about that. And we just wanted to touch on it briefly tonight. I think that if you're part of the Beekeepers of Indiana, you probably saw his obituary circulated. And there was a brief comment, I think, in the most recent newsletter. But Barry was one of the founding members of this club and part of the core team since its inception. He's very active. He was very generous with this club, both with his resources and his time. He helped to lead the club. He spent a lot of time working with new beekeepers. He was a very active beekeeper, both in Hamilton County here and down in Switzerland County, where he and his wife Ann have some property. He was the operator of Lost Fork Valley Farms, or Lost Fork Valley Honey. And anyway, we as a group, obviously, we can't begin to talk about what his loss has meant to us. It's been huge. I know personally, I'm still trying to come to grips with that, and we as a group are going to be trying to come up with a way that we can create some fitting tribute or memorial to him for all he meant to this club and to the beekeepers in the state of Indiana. But we just wanted to make everyone aware of the fact that he did die last month, that it was very unexpected, and that he is greatly missed. So thank you.
Okay, I think I'm next up. Good evening, all. Thank you for joining us. My name is Jeanette Wiles, and I find that at our meetings that we just have our core group just sort of starts talking, and you don't know who they are or anything about them. Yeah, you start talking, Jim. So I want to introduce myself. I actually wear several hats around here. I'm part of the core group. And I'm coordinator for the Jump Starter program as of recently. Also, I'm a director at large at the state organization, the Beekeepers of Indiana, where I'm on the scholarship committee, the education committee, and I'm chairperson for the Young Beekeepers Awards program. I'm also a Purdue Master Gardener and often give flower-related presentations to this club and to some others. Tonight, I'd like to do a couple of ads first for the programs with which I'm directly involved. You can find information on both of them at the Beekeepers of Indiana website. And I'm going to try to share my screen. Here we go. Paul, if you can make me a presenter or whatever.
Uno momento.
Yeah, it's coming. There we go. Okay. Okay, the first thing is if you go to indianabeekeeper.com, this is where several things that I'm going to talk about are. The first thing is it looks like this when you get in there, and it doesn't have this little orange arrow up there pointing to program. But if you click on that tab, you get a drop box, and then you click on what you want from there. You can get the information about the program from that. It goes to this page, talks about the program. If you are interested, if you know somebody who might be interested, be sure to tell them about this. Has to be somebody 12 to 22 years old. And if you click on this, it'll take you to the application. And notice these awards that are at the top. They're quite generous. So if you know somebody who fits into this age group, this is a great thing. And oftentimes we don't have enough applicants to take all the money. So think about that, look hard. If you know some 4-H'ers, it's open to 4-H. It's not restricted to our club or to the Beekeepers of Indiana. Okay, the next program is the Jump Starter program. Again, you go to the programs, click on Jump Starter, and it'll bring you here. And this gives you all the rules for it. The application is not online because this award goes through our club. Only one applicant per club is allowed. If you're interested in this, contact any core member or me, and we'll talk to you about getting it set up. As I said, there's only one per year. So in some cases, we have to make decisions. This year, our Jump Starter winner is Jessica Benz, and her mentor is Kim Patterson. So we'll be looking for somebody for next year. Bee School 19 is February 27th at the Horizon Conference Center in Muncie. It's being run very differently this year due to COVID regulations. As Tracy said, it's limited to 250 attendees, 50 of whom will be beginners. Okay, then normally we have around 1,200 people, 200 of which are beginners. So if you want to go, be sure to get online and send in your registration. Now, it's a little bit different because what they're having people do is you sign up for one track or a location, and you stay in that classroom all day, and the instructors come to you so that they don't have people milling around in the halls and that sort of thing. They feel that it's safer this way. So what you need to do is when you go online, this is actually as part of the registration online, and you see which group of classes you would like to go to. As you can see there, some of them repeat and some of them are different. So you choose the one that suits you the best. And on your registration, you tell them which one you want. As I said, that's very different from what we usually do, but because of COVID, we just can't have people moving around so much. Okay, now back to our regular programming. What's blooming in January is nothing. So if you're thinking of gardening for your bees though, and it's a good time to think about it, because if you are thinking about starting seeds for gardening or anything like that, now's the time to be doing it. And what to plant, the best place that I have found is our state apiarist, Kathleen Pro, wrote an article, and it's on the DNR website. I find the best way to get to it is to go to the in.gov/DNR and then do a search either for her name or for gardening for honeybees. She has lists there of trees that are good for honeybees, shrubs, perennials, annuals, and it's quite a comprehensive list. So you can, as you're planting, you can kind of aim towards planting things for your bees. Now, you don't have to have a large area to do a full bee pasture or anything. Every flower helps. Okay, just like there are endangered animals, there are endangered plants in Hamilton County too. Over the state, there's a huge list of endangered plants. They narrow it down into Hamilton County to just 6 of them, which I'll talk about very briefly. The first one is rose turtlehead. Rose turtlehead likes wet areas. It is attractive to bees, and it blooms in the fall. The next one is Virginia sneezeweed. Now, there is another sneezeweed that's a common sneezeweed that you see all over the place. This is a very specific one. So it's bee friendly, and it's also late fall blooming. Then this pretty little white thing over here on the right is a prairie white fringed orchid. It likes damp areas and not as wet as rose turtle head, but it does like damp areas. It blooms in June and July. Due to the structure of the flowers, though, it's really not bee friendly. They really can't get down in there, just like other orchids. The cucumber magnolia is a 60 to 75 foot tall tree. If you look at this bloom that I have up here, it probably reminds you of our tulip tree. And it is a first cousin to the tulip tree. Our tulip is commonly called a tulip poplar. It is absolutely not a poplar. It's a magnolia like this. Lake cress is an aquatic plant. You can see it here growing in the pond. It blooms in June to August, has little pollination, generally grows by underwater vegetative reproduction. So it just sends out new shoots underwater. It doesn't, the flowers are not attractive to bees. And then the last one is a fun plant. It's called spoon leaf sundew. This is adamantly not bee friendly. It is an insectivore. It eats insects. It has very sticky leaves. And the reason actually that these plants have developed, the insectivore plants have developed to eat insects is that typically, like this one, they grow in areas where there's very low soil nutrients. So they need something to feed on. So they catch these insects, eat them, and that's their nutrition instead of getting it out of the soil. Now, note that these are specific varieties of what might be a more common plant. The rose turtlehead and the Virginia sneezeweed are examples of that. They are very specific. One of the reasons that these plants are endangered is that they are mostly very attractive looking. Oh, by the way, the cucumber magnolia, you'll probably never see blooming. As I said, it's a 60 to 75 foot tall tree. It only blooms at the top. The leaves, though, are about 10 to 15 inches long. So one of the reasons that these plants are endangered, at least the little plants, are that people have removed them from their location. They think they'll take them home and, take care of them there and grow them. These plants, however, are very picky about their environment and generally succumb no matter how much care they're given. So leave them be if you see them. In addition to that, it is illegal to move them. Some of them you can find in some nurseries, not like the big box stores, but in some specific nurseries. So if you want to buy one, the question is, what are you going to do with it in your garden? So I found an idea for that. And that's what I have for the night.
Thanks, Jeanette.
That was good. That was good.
Now, the name of our next presenter was given to me as Garret Slater, but I'm seeing a different Slater.
Yes, it's Garret.
Garret.
Yep.
Welcome, Garret.
Let me share this screen.
Jeanette, can you stop sharing, please?
I'm trying to find out how to do that. I'm sorry. Let's see if
I got you.
Did you get me?
Yep.
Okay, good. Thank you. There we go.
Okay, how does that look? Okay. Well, cool. I'm really excited to give you guys a talk today. I know I was supposed to give a talk a while ago with COVID hit, but it's really fun to finally give talks to beekeepers again. So really excited to kind of present like what I've been working on and things I plan on doing in the future. So just this presentation based on we're talking about drones. When you guys think about healthy colony, do you guys think about drones? Any raise of hands? Yeah, not many people. Oh, one person does. Great. Yeah. So drones are really important, but we really don't think about them when you think about colony health and even queen quality. So during my presentation, I'll talk about them. But before that, I kind of want to give a little background about myself. So I really got interested in drones when I started beekeeping. So I'm from a rural town in North Dakota. I grew up in a commercial beekeeping family. My uncle he ran 10,000 colonies or so, and I started working for another beekeeper that had about 50,000 colonies. So I come from like a really commercialized keeping me. And when I started working with them, especially when I turned 14, the one thing I really became interested in was queen failure. We'd buy queens from different sources across the country. We'd buy them from Georgia, California. We'd buy them from Texas, and these queens would be failing within 6 months. And question was, why are these queens failing? Because really what you want to do, you want colonies to build up, you want them to be strong, going into the honey flow, and these colonies are failing beforehand. So that's what really got me interested in queen failure and what are the reasons. If you look at these surveys, the NAS survey and the BIP survey, queen failure is the second largest issue reported by beekeepers. So I don't know if you guys experience queen failure. I'm sure maybe you guys are rearing your own queens, so you maybe have less issues, but this is a big issue among beekeepers across the country. So this is something kind of a central question in my research was, why do queens fail? So yeah, and here's a graphic showing the colony losses for the Bee Informed Partnership. Just seeing the massive losses that beekeepers experience across the country are up to 30 to 40%, which is just astronomical. And does queen failure play a role in that? So yeah, like I said, I'm from North Dakota. Can you see the cursor? You guys can see the cursor if I'm pointing to it?
Yes.
Okay. So yeah, North Dakota has 700,000 registered colonies, which pretty much outnumber the number of people in North Dakota. So it's a really there's a ton of colonies in North Dakota, and it's almost too much, but it's a perfect area for bees because there's a lot of wildflower, there's a lot of commercial crops such as canola that can really support all these colonies. But then again, you have a lot of other issues that come around with that, and that's what's really nice about coming to Indiana is. There's not nearly as many beekeepers, and your honey flow is it's a very long honey flow season, which is great, really from May until, August. But yeah, once again, I experienced queen failure in North Dakota, and that's really spearheaded my interest in, queens. And so after that, I went to North Dakota State University in Fargo, and I looked at, how's nutrition contributing to queen quality, and I was focused on royal jelly, and if I change royal jelly content, how did that affect queens? And as you can see, if I changed the food that I fed to the workers, it actually changed the queen development. So this is the queen, this is the worker, and this is the intercaste. And what's really interesting is if you change nutrition, these individuals are actually kind of half workers, half queens. So they're kind of that in between. And if we did a study where we surveyed queens across North America that were failing, we see that some queens were actually less developed. So they're actually kind of more of an intercaste than a true queen. So that was something I did in my master's degree. And after that, I started working for the Bee Informed Partnership for Marla Spivak in the Midwest team. And basically, it's really extension heavy role where I worked with about 30 to 40 beekeepers across the region from Minnesota to Wisconsin to North Dakota. And this was like a really great experience because I worked with so many different beekeepers. They, a lot of them are third, fourth generation beekeepers, and they did a lot of things differently. They did things that, I probably wouldn't agree with, but it was a really cool learning experience from them because, these people are professionals at beekeeping. But one thing I also observed was, queen failure was still an issue. And what was really interesting is some beekeepers would try to, they try to rear their own queens. Hopefully that you try to fix that issue, or they buy queens from a lot of different sources. So from one source, such as a California source, experienced queen failure, they had something to kind of fall back on. But was that sustainable? I don't know. So because of that, I decided to pursue a PhD and kind of focus on not on the queen side of it, but on the drone side of it, because there definitely is a lot of research on queen failure from the queen side of it, but we really don't think about the drones really, because the drones are providing the sperm. They are providing these material benefits that actually impact queen fertility and colony health. So yeah, so I found Brock and I started, pursuing a PhD with Brock Harper, focusing on drones. And yeah, here's some really cool photos of drone mating and then insemination, which I'll talk about a little bit during the presentation. Okay, so a little background about drones. So I'm not sure where your club is. I'm assuming some are beginners, some are more advanced beekeepers, but I'm kind of give you like a general background of drones. So drones are a result of an unfertilized egg. So queen, she'll fertilize the egg, and those will become a worker, or they can develop into a queen. But she'll also lay eggs that are unfertilized, and these become haploid eggs. And these haploid eggs only have one set of chromosomes, whereas the females have two sets of chromosomes. And so these drones will develop in a colony, and they're taken care of by the nurse. And I want you to think about this way, that the social colonies actually, they're regulating the nutrition of the drones. They control the drones during development and then during adult. So once the drones emerge, the workers are actually feeding the drones really for the first 7 to 10 days. And this is really unique compared to a lot of other insects, other taxons, where once the males emerge, such as other bees, these males eventually start mating. But in the honeybee colony, the colony actually takes care of the drones. They feed them, they take care of them, and they make sure that their basically that their reproduction is maximized. And so around 7 to 10 days, the drones mature. And what that basically means is that the sperm is produced during development, during the pupation stage, but the sperm actually doesn't migrate or become mature until around day 7 or 8. And the sperm is actually migrating down the endophallus to the tip right here. So this kind of shows that this drone is mature. And if a drone wasn't mature, so I don't know if you guys have ever taken a drone and kind of squeeze them and had their endophallus pop out. This brown speck at the end, if that happens, that means this drone's mature. But oftentimes you'll get a drone in a colony and it'll be kind of white at the end, or there won't be much. And that basically means the drone is not mature. So around 7 to 10 days, the drone is probably mature, and they fly to these drone congregation areas. And if you ever get a chance to look at these congregation areas, they're really cool sight. I'm sure you can find some YouTube videos on them, but these areas have thousands and thousands of drones, and some estimates say around 20,000 drones. And these drones basically go to these areas day in, day out, and they fly around just waiting for a queen to arrive. And if a queen does arrive, they produce these really cool male bias comets, where these drones are just chasing the queen in the air, trying to inseminate the queen. And the queen will, the drone does latch on. The drone will die and then fall down, and the queen will continue to mate, and she'll mate with around 15 to 20 males. So, and this is known as the flight of the Virgin, and it's a really cool sight. If you ever see it in person or watch a YouTube video of it, it's really cool. But it's a very competitive sight, right? Because if there's one queen and 20,000 males, these males are competing really aggressively against each other to actually inseminate the queen. So let's say a male is successful, and the queen mates with 16 of these 20,000 drones. So the competition doesn't end there. The queen actually only accepts 4.7 million sperm. And just to give that a little context, a typical male probably produces about 4 million sperm cells. So she's only accepting like 1/16 of that sperm. So even though a drone may actually mate with the queen, doesn't necessarily mean that he's successful. And then after that, the queen, she'll store 4.7 million sperm cells, but she'll only actually use 1.6 million sperm cells throughout her life if she lives 2 to 3 years. So this whole process is very, very competitive for a male, where there's this extreme competition at these congregation areas, but then also sperm competition, really to actually get sperm stored and actually get utilized. So really, if there was a low quality drone, that's going to impact its ability to actually fertilize the egg further on. So I hope that makes sense. If you have more questions about drone biology, it's really interesting, and we really don't know a lot about it. We're learning a lot more, and really, research have really ignored drones. They focus on queens or other parts of colony health, but drone health is largely ignored. Let's see the next one. Okay, so So why should we focus on drones? Are drones important for queen quality? So I don't know if you ever have read this study. This study by Jeffrey Pettis in 2016, and he was observing queen failure, and beekeepers were telling about queen failure. So what he did was he had beekeepers send in failing queens from across the country. So these are queens from his ARS in Beltsville, Maryland, and then he got queens from the east, queens from the west, and then queens that they must have purchased and they requeened. And they looked at sperm viability. And basically what sperm viability does, it looks at what percentage of the sperm that the male is producing is actually alive versus dead. And typically this is what you should expect. Most drones are producing sperm that's about 90% alive. There is some variation where it's 80% all the way up to 100%, but it's around this 80 to 100%. But these failing queens, their sperm that they're storing in the spermatheca was really around 60 to even as low as 40%. Now, if you can imagine, I mean, that's really low. And if a queen is trying to produce workers, it's possible that you might see a spotty brood pattern, right? So if the sperm isn't viable, it's not going to successfully fertilize the egg. If it's not viable, you're going to see a really spotty brood pattern, which is what these beekeepers are seeing, and it's likely linked to sperm viability. Now, you can view these results in a few different ways. Is it the queen issue, where is the queen having issue actually storing the sperm? And there's a lot of really cool studies on that recently, but it could also be to the drone part. So if the drone is only producing sperm that's 40, 60% viable, then that could be one of the leading causes to queen failure. And so I did a little bit digging deeper, and this is a researcher down in Texas in Juliana Rangel's lab, and she does some work with drone sperm, and they had sort of the same question: how high quality are these drones in commercial operations in Texas? So she went to this graduate student. Adrian, she went to different locations in Texas and sampled drones, and she looked at the sperm viability. So how viable are these sperm? And she's seeing, like, look at this range in these colonies. Like some drones had sperm viability like 10%, whereas some were 100%. And this is a massive variation. So you see that, I mean, these colonies are producing drones that are producing very, very low quality sperm. And this could be due to a lot of different reasons. I mean, the high uses of amitraz, insecticides, antibiotics, nutrition, anything like that. But regardless of that, we're seeing that sperm viability is really low, and this is probably linked to queen failure. And so drones are very sensitive to the environment, and one reason that the sperm viability may be very low is due to many different things, such as miticides. So amitraz is known to have a huge effect on sperm viability. As you can see here, sperm viability from 0 to 100, the control versus drones that are treated with amitraz, it knocked their viability down 20%. We also see another study, thymol, coumaphosphofluvalinate, which beekeepers aren't really using anymore, but I mean, those had a huge effect back in the early 2000s, late 90s. We see field chemicals such as fungicides, insecticides, fipronil, which is some of the beekeepers use for small hive beetle. It's basically a roach insecticide. Can have a huge effect on sperm viability. So, all these different environmental factors, are definitely impacting the drones and could be linked to queen failure. So we know that there are these factors that are impacting, drone quality. But what about the genetics? So are genetics playing a role? So, there's a lot of studies for people looking at the environment part of it, but what about genetics? So this was a study, and this looked at sperm viability. So 100%, that means 100% of the sperm is viable versus 0.5 is only 50%. And this is kind of confusing, but just look at each line as a different distinct breeding line. So basically, this is 5 different breeding lines where beekeepers are basically selecting for their own traits. So some beekeepers are selecting for hygienic behavior, some are selecting for other traits. And as you can see, the viability differ quite a bit between these different genetic lines. So if a beekeeper is selecting for a trait, they're not only selecting for the trait of interest, such as maybe pollen hoarding or hygienic behavior, but they're also indirectly selecting for these male traits. So it does seem that there is a genetic component to this variation. And because, we are a part of a genetics lab, one of my questions was, what are the genes that shape these traits? Because one thing that we would want beekeepers to do, if this is really important for queen quality, is we'd want beekeepers to eventually select for drone traits to improve drone quality and maybe reduce their sensitivity to environmental components, things like that. So during this presentation, I'm talking about the genetics of drones, and one thing is what shapes a male trait. So here is a male drone, and here is its sperm, and here is its testes, and everything that is involved with reproduction. And my goal is to really fulfill this whole thing. So getting from the genetic components, right, the genotype, and how that interacts with the environment, and how that basically that combination between the environment and the genotype shape these traits. So first I'm going to focus on the genotypes. Don't worry about the external environment. Just focus on the genetic part and how that shapes the trait of interest. So I started about a few years ago. So I've had 2 summers doing this research, and basically I'm looking at 2 important points. So I have a colony, and they go to these drone congregation areas, and then if they're successful, they mate with the queen, right? So I'm looking at 2 important points. So traits at these congregation areas, and then traits at the actual insemination point. So for the important point 1, I'm looking at flight traits. So basically what I did was I put these RFID tags on the drones, and they're very, very small, and they fit on the back of the drone. And basically I just use like this glue. I put the tag on, and I put it into this colony. And in front of the colony, it has this sensor, so it can track when a drone leaves and comes back to the colony. And it's really cool. So it doesn't tell me necessarily the mating behavior, but it tells me the flight behavior. So it tells me when it leaves, how long it's gone for, how many flights it's doing, when it actually begins flying. So I can answer a lot of cool questions using these RFID tags. I will tell you, it takes a really long time to mark 1,000 drones. I spent some long nights, painting them all. Fortunately, they're big, and they don't sting, so they made the job a little bit easier. And then on to important point 2 is I basically collected drones at different time points from age 10 all the way to age 21, and I looked at important sperm traits. So I looked at how viable the sperm was, how long the sperm was, how motile the sperm was, and things beekeepers would be really interested in traits that would be important for queen fertility. So just think about important point 1 at these drone congregation areas, and point number 2 where the actual sperm competition is occurring at. And just another point, this is what a mature drone looks like and a non-mature drone looks like. People that probably have experience with beekeeping, they've probably seen this where they grab a drone and just squeeze them, and it doesn't produce anything. This basically shows that the drone is not mature, whereas this one is mature. And yeah, this is a really cool phenomenon in honeybees. Okay, and so to do this experiment, so I collected 3 unique breeding populations across the country, and I chose these populations based on genetic background, based on breeding. And so one was the Purdue mite biters. So we've been selected for mite biting behavior for about 10, 15, 20 years, something like that. So I used that one, and then 2 other unique breeding populations, because basically what I wanted was I wanted 3 unique breeding lines that had variation in male reproductive traits. So I did a common garden experiment where I had these 3 breeding lines, and I brought them to a common place in Indiana so I can control for all the environmental factors. And I marked drones using the RFID tags. So this is what it looks like. And I looked at the flight behavior, and I also looked at sperm traits. So that's what the RFID tag looks like. And basically, my question was, how do breeding populations vary? So looking at flight behavior and looking at sperm traits. And I won't talk about them all today because I'm still kind of analyzing the data, but I do have some results on the flight data and also on the sperm results. So here are my 2 questions. Does reproductive senescence vary? And when we say senescence, I mean aging. So does it vary when they become mature? And does it vary how the sperm changes over time? So based on other studies, the viability should decrease over time, but does that vary between lines? So that was one major question. The second major question was, does mating quality vary? So are they producing different amounts of sperm, and are the sperm different in viability? And what's really cool about this experiment is basically I'm controlling for everything. I'm not using I'm not using the chemicals from mites that are going to be hurtful to the drones. So basically, I'm trying to reduce that amount, at least during the experiments. I'm using like salicylic acid beforehand and afterhand, and I'm making sure that there's nothing on the outside that could be affecting drones. It's all due to genetics. So that's really cool about this common garden experiment is I'm controlling for all those factors that are known to affect drones. And the only factor that should cause variation is the actual genetics. Yep, and these are flights I looked at. So number of flights per day, flight time of day, first flight duration. So with these RFID tags, I was able to look at about 50 different flight phenotypes. So I was kind of figuring out which phenotypes are important, which ones kind of covariate with each other. So these ones I looked at. And here's the preliminary results. So line 1, 2, and 3. So line 1's in the red, line 2 is in the black, line 3 is in the blue. And this looks at the density of flights and how long they're flying. So are they flying 0 to 80 minutes? And at what point are they flying the most? So as you can see, the drones from the black line, they're actually on these really short, rapid flights. So they're only flying for 0 to 20 minutes, really, for most of their flights. Whereas, line 3, they have this mixture between these short flights and these longer flights. Yeah, so it's really interesting. It only tells me flight information, but if we think about how this maybe connects to mating information, this could connect to where they're going for DCA. So maybe these drones are flying to DCAs that are farther away, whereas these drones are flying ones that are closer away. Or maybe there's some kind of behavior where, with workers, where they're kind of searching for the DCA. Maybe these rapid flights mean they're kind of searching for these DCAs, trying to find the right DCA, or like maybe the sexiest DCA, and going to those locations. So yeah, this is some really cool variation where some lines are flying for very short periods of time, whereas other lines are flying for longer periods of time. And I'm also looking at other flights, but this is kind of the most interesting result so far. So next I looked at age of first flight and the number of flights per day. So basically what I was telling you was drones typically don't fly until they reach maturity. And, really when you read any type of, book on bees, they say that's around 8 to 10 days. And we see there's actually a lot of variation to that. So for line 1, 2, and 3, line 1 really didn't begin flying until age 8, whereas line 3 began flying at age 6. So they actually began flying 2 days earlier, which is a lot more variation I really expected. I thought they'd be flying around the same time because they are in the same colonies. So you'd think that maybe the workers would be regulating when they fly, but it seems that there may be a genetic component to when they actually begin to fly. Next is flights per day. So how many flights are they taking per day? And you see line 1, they're taking about 10 flights per day. Line 2, 8 flights per day, and line 3, around 6 flights per day. So there's definitely a lot of variation in the number of flights that these drones are taking per day. So, the first big takeaway here is that there is a genetic component to this first trait, flight, that there's differences in how long they fly, when they begin flying, and how many flights they take per day. So the second main point is looking at the sperm traits. So these are sperm traits I looked at. So I looked at sperm concentration, viability, motility, so how much is the sperm moving, mitochondrial activity, so how much energy are they producing, throughout their lifespan, and then other morphology traits. And so the first one I looked at was just simply maturity. Were these drones mature? And I looked at 2 different ages, age 7 and age 14. And once again, this is what a drone looks like when it's mature. When you squeeze it, there's the brown solution sort of at the tip. And if you squeeze them and there isn't, it's clear like this. So the sperm hasn't fully graded down. So we look at line 1, 2, and 3. We see at line 1, about 40% were actually mature. Line 2 and 3, only 10% were mature. So it seems like line 2 and 3 are maturing slower. And if you look at age 14, we see a very similar trend. But it's really interesting that at age 14 that they weren't all mature, only 60% were mature. So I thought, and for these 2 lines, I mean, only 20 to 30% were actually mature. So, really from what I've read, age 15 should be the point where they're just maximizing their fertility. This is when they should be, fully mature and ready to actually inseminate a queen. But in fact, we're seeing that a large proportion of the drones actually are not mature. So yeah, and this is a really interesting question I'd like to look more into, and it's really important for queen breeders or people that want to do artificial insemination, because they really want drones to be mature. So trying to figure out why these drones are maturing faster than they should be. Next, I looked at sperm traits. So the 2 sperm traits, sperm concentration. So basically, how much sperm are these drones producing? And we see that line 1, they're actually producing only about 1 to 2 million sperm cells, whereas line 3, they're producing 4 million sperm cells. And so what's really interesting, if you look back beforehand, we see that the line 1, right, they began flying earlier, they began flying more vigorously, they had these sort of longer flights. And so there may be a trade-off to flying, right? So maybe if you're flying more aggressively, you're flying earlier, that could affect, the sperm traits, because, and the colony is long, the workers aren't taking care of you, you're exposing yourself to an environment earlier on. So, that might explain why, the sperm concentration was very low for line 1 versus line 2 and 3, because, these colonies weren't in line 2 and 3, these drones weren't flying as long or as vigorously. So, I looked to look more into the trade-off between that, because I think it's really interesting. Next, I looked at viability. So what percentage of sperm was actually alive? And I looked at, just age 7. I don't have the results for age 14 and 21 in this presentation, but the results are very, very similar. So I just showed age 7. And we see that line 1 and 2, their viability was only around 50% viable, and line 3 was around 80%. So, over time, at age 14, the viability went up to around 80%, so it became more normal. But the sperm didn't become viable right away. It took longer to become viable. So really the big takeaway here is, these were all raised in the same colony, but we see like a ton of variation, like really a lot more than I thought these would. And these are basically just breeding populations from across the country, from beekeepers that are selecting for what they're very interested in. And we see like a tremendous amount of variation. So really cool, interesting results so far. And it shows that there is a genetic component to male reproductive traits. And yeah, and basically, I'm not showing this stuff in the presentation, but I looked at other traits such as size, morphology, wing morphology, and then I'm looking at the RNA also. So back to the really the major conclusions of the presentation. I looked at 3 distinct breeding lines, and I looked at traits at 2 important time points. Number 1, at these drone congregation areas, and then number 2, the sperm related traits. So at number 1, I found that drones begin flight at different ages. Drones also fly at different times. They fly longer versus shorter flights, and there's just a ton of variation in these flight phenotypes. As for the second important point, drones mature at different rates. So some drones are maturing faster, whereas some drones are maturing slower. And there could be a trade-off with maturity and sperm production with the ability to fly. So if drones are flying earlier, that could also affect maturity and concentration. So these traits are interconnected, but they're also really important for beekeepers. So future directions. So I'm going to finish the phenotyping and characterize this variation. So I definitely have a lot more traits to look at, and I'm almost finished with that. I'm going to try to characterize the variation. Next, I have these genotypes. So I looked at their DNA, and next is to associate which genetic markers are associated with this phenotype. And so can I find maybe some markers are associated with slower maturity, maybe some markers are associated with higher sperm production. Which traits, which genes are important for these male reproductive traits? And then, if I can identify these markers associated with these key fertility traits, we can hopefully use these markers to help beekeepers select for these traits. Because, if you're a beekeeper, are you going to be out there using RFID tags, selecting for flight behavior, or are you going to be looking at the sperm traits? There's no reasonable way to do that for a beekeeper, especially on a commercial level where they're raising queens. So how can we, identify markers that are associated with the fertility traits so beekeepers can select for them? And basically, yeah, so that's really the first part of the project. I'm going to finish that hopefully the end of this year, but this next summer I'm going to integrate the environment into this equation. So I talked about the genotype all the way to the phenotype, but how does the genotype interact with the environment, and how does that shape these traits? Because these are in one single environment, but what happens if I change the environment? Would that affect the trait positively or negatively? So this next the next summer, I'm going to be spreading my colonies across Indiana. So I'm looking for locations probably in the southern part, the central part, and the northern part to see how really the environment interacts with the genotype and how that shapes these traits. So if you guys have any locations, know any beekeepers that are interested in kind of collaborating and helping me out, I'm still kind of looking for locations. So I have the central part covered, but I still need beekeepers to work with in the south versus the north. So if anybody be interested in this project, I think it'd be really kind of a fun collaboration. And really, so my long-term goal is to produce like a genetic test for beekeepers. So I'm sure a lot of you have taken like a 23andMe test or ancestry test, and basically it's a test that kind of shows you how likely you are to have a trait or not. And for me, I think it's a really cool concept for a breeder. If you could do a genetic test on maybe the colony and tell them, are they more likely to have hygienic behavior? Are they more likely to have high quality drones, high quality queens, things like that? And it'd make it sort of easier for queen producer to select for colonies that are high quality. And eventually this test is going to be really cheap and to be easy for beavers to select for. So part of my project is really looking at the drone angle. So how can we find markers associated with male reproductive traits, and using a format where we can help beekeepers actually select for this trait in an easy and cost-effective way. So that's one of the long-term goals of mine, and one of the long-term goals of Dr. Brock Harper at the lab. So we're looking at a lot of important industry-related traits, and mine is really looking at the drone part of it. Yep, like I said, so next up is drone, so looking at the male reproductive traits. So, and hopefully I can kind of link this up to why do queens fail? This is a question that I've always been interested in, and I really think that drones play a major part of this. Obviously, it's not the full equation, and maybe queen failure is, obviously isn't the largest cause of colony death, but it's a major contributor, and it does impact colony health and production, which is important for beekeepers across the country. So, hopefully we can sort of solve this issue, or kind of find out maybe sustainable solutions for beekeepers, to figure out how to deal with this issue. So with that, I still have a lot. I just started like 1 summer ago, so I'm still kind of my preliminary data. Hopefully by the next couple of years, maybe you have me back again, I can present kind of the full set of data. But I think so far we're showing some really interesting results that there is a ton of variation in these drones, and there is a genetic component to it, and something that beekeepers can select for. So with that, I'll thank my funding sources: the Foundation for the Preservation of Honeybees, Project APISM, and the North Dakota Department of Ag. And then my advisor, you guys know Crispin, he's helped me a lot with the project. I talk about the insemination part of it, but there's insemination that's really key to this project, and he's just the best at it. And then my lab mates and people that help me with the project. So, with that, I'd probably take any questions. I can talk more about drone biology or my project, or I'm really open to anything. So.
And Garrett, I don't know that you saw, but we had one of our members has volunteered to be in your study from the southern Indiana area.
Oh, perfect. Yeah, I don't know any beekeepers down there, so I'm really new to the area. So yeah, hopefully we can connect.
I'll be able to get you guys to exchange some information later on.
Perfect. Okay, looks like we have quite a few people in here. Okay.
Yep. Garrett, we had a question from someone in the chat asking if the colonies that you were working with in your study, are they all of a particular race, or have you looked at the racial component?
Yes, that's an excellent question. So we're actually just getting results in, and so what we did was we looked at what is the genetic background of these 3 ones, and we chose them based on the genetic background. So one has a little bit of A lineage, just a small set of it. So that's Africanized lineage, and has mostly M and C, which are mellifera and carnica. The other one has mostly M, and the other one has mostly C. So because of that, we have kind of a wide range of colonies with distinct genetic backgrounds. And yeah, and I'm really glad, because I wasn't sure if they would or not, because basically I kind of randomly selected them, but I was hoping they'd have a pretty diverse background and genetic lineage. So, one breeding line's from the southern part of the United States, one's from the western part, and then one's from Indiana. So, I was hoping I kind of capture all the variation that there is in the United States. And the Maddie, she's in a project where she's actually genotyping all the colonies, and she found that there is a lot of variation in the genetic background of these colonies. So she's working on that, and hopefully we can send it to you guys and you see how much variation there is between the colonies I'm using. But thankful there is, because that probably explains why I'm seeing so much variation, because they are coming from different parts. And just to note with that, there's some really cool studies looking at bees, like Africanized bees versus Asian bees, for example, and they see that they fly at different times, they fly at different heights. These congregation areas are different areas, so there is a lot of variation in those, and that probably explains why I'm seeing so much variation with my colonies.
Hey, Garrett, this is Thomas. I was curious because I was wondering if having more drones also in your hive is advantageous to the hive in regards to less mites and less pests, or less mites rather.
So if
you know anything about that?
So if you had more drones, there'd be less mites.
Yes.
I would think it'd actually be the opposite. So
okay.
Yeah, so just with raising drones, I don't know if you guys have ever put drone combs in your colony. It's just a magnet for mites. So it's been really difficult, because basically I'm putting these drone combs in these colonies and it's just like reproducing mites. So I've had to kind of work around that. So basically once the drones emerge, well, I'm basically drone emerging, they have a mite. I just throw that out and then put those drones without the mites into the colony. But those colonies where I'm raising drones, it's just a mess with mites. So it just had to, what I've been doing is just basically freezing some of the drone frames just to kill some of the mites, and that works pretty well.
Do we have any more questions?
Was there anything on the biology? I wasn't sure if I covered that too quickly.
Garrett, I have a question. It's Jeanette. As I was looking through this, it appeared that your colony 1 flew earlier, but the sperm was not mature. Is that correct?
Yes. Yeah, let me go back to that. So that was one of the key findings. So let's open that back up. Yep. So line 1 started flying earlier, and they look at the sperm traits. Maturity was higher, but the concentration was lower.
Yeah, that's interesting.
Yeah, so it seems like there is a trade-off where these drones are flying at an earlier time, and that could be due to where I got the colonies, right? They're from maybe a warmer part of the United States, that they may be flying earlier because of that, because the weather's different, but that could be affecting their sperm negatively. So there seems to be some sort of trade-off with flight time and the amount of sperm and the viable sperm that they're producing. Okay, so what is the failure rate of AI queens versus open mated queens? Yeah, that's a really good question. I think it's for AI queens. I think it depends on how well you can inseminate. I don't have a lot of experience with that, but I think with Crispin, I AI'd about 40 to 50 queens, and I think we had a really good success with those. But the problem is, they typically don't live as long, so they tend to fail within a year and a half or so. Whereas open mated queens, it depends where you buy them. I bought in queens from all over. When I was a commercial beekeeper, some from California, Texas, Florida, and it didn't. The region really matter that much. Sometimes you just get a batch of queens that would just fail really quickly. So it's tough to say. What causes that could be due to weather. So if it's a really rainy day and the queens will have the opportunity to really successfully mate. So, maybe they're in Texas and it's raining for the 2 weeks. Those queens are probably more likely to fail because of that, because weather related things, due to, wide range of things. So yeah, it's tough to answer that question, but I'd say like a successfully open mated queen, they're probably going to fail maybe 2 to 3 years versus AI queens probably going to fail within a year, year and a half. If you're good at actually artificial insemination. How do the Jones know where the congregation area is in Queens 2? Yeah, that's a really good question. That's something I've always been intrigued by, and that's something we just don't know much about. So Howard Jones, first of all, like where they find these congregation areas, and how do they know where these congregation areas are? So how do they come in and out like this? And it makes me think that there's some kind of waggle dance involved, but I don't know. It's never been studied. But what I noticed, and I didn't show this in this presentation, was Jones when they're younger, they do these really short, rapid flights, really for the first 2 to 3 days. So it makes you think that they're trying to like kind of scout out kind of a congregation area to go to, and then at the end they're going for these longer flights. And that could be due to maybe when they're older, they don't have like a lot of time left to mate, so they're spending more time outside the colony. They can probably fly longer too because they're more mature, and maybe they're staying in the congregation areas longer. But the actual cues is kind of relatively unknown. We do know that the Jones produce a pheromone that seems to attract Jones. So I've isolated this pheromone and I've put it up into the air at the bee lab, and it does seem to attract other Jones. So the Jones do seem to be able to probably sense when other Jones are in the area, but how they're choosing these areas is I'm not really quite sure. A lot of people kind of they looked at like structures, like landscape structures of how they're choosing these areas, and it seems that they're going to kind of down in a valley next to trees. So they're going to these open areas, but it's next to like a landmark, like a tree, or even like stadiums. Stadiums is a really hot spot to find congregation areas. So yeah, we're still learning a lot more about how they're finding it. As for queens, yeah, that's a really interesting question. So I'm not quite sure. There's definitely a lot of debate around whether congregation areas are actually a thing, or if drones are just randomly flying and maybe randomly encounter a drone. But the one thing we do know is drones tend to fly farther away from the colony, whereas queens tend to mate around like kind of closer to the colony. So that might be a way to avoid inbreeding for the colonies. So we do know that part. So the queens are probably flying maybe half mile up to a mile, whereas Jones may be flying up to 2 to 3 miles. I mean, just from a very few studies that people have actually looked at that, but that's one thing we know, and I think there's a lot more information that we can learn from, where these congregation areas and how they find them. And is there such a thing as a DCA that's set up and stocked with selected drones? Yeah, that's a really that's probably ideal, right? If you could just make a DCA, if you could find the DCA and just stock with the drones of your interests, and you would worry about the insemination, that would definitely be the dream. And maybe I've kind of thought about this. If you could find the pheromone where you could sort of maybe attract the drones and set up like maybe drone colonies. I've read in some books where you can set up colonies that just produce a ton of drones, and maybe you can set them into these areas where you found a DCA. So maybe if you were able to find a DCA, I'm not sure how you find them. I haven't been able to find one. So if you guys can find one, good luck. But if you can find one, maybe put colonies that you're interested in there, and to put like a really strong pheromone to attract those drones so they don't fly away. That could be one way to do it. But yeah, that'd be the ideal situation if you could actually do that. But really finding the DCAs is really difficult. And I've been trying for the last couple of years. I put like a queen on a stick walking around. I've used isolated pheromones, but I just haven't been able to. So I think the next thing to do is use like an actual mechanical drone, that flies around the pheromone. That might be the easiest way. I don't know. Okay, I got your email, Bruce. I'm gonna reach out to you. Thank you for helping me out, guys. And
Sure. Any more questions for Garret?
I'd like to say that I think his comment, "Queen on the stick," has got to be my favorite thing that he said tonight.
I'm glad I'm one good takeaway. Maybe it's like a new fair food, I guess.
Well, Garret, thanks so much for taking the time to come and speak to us tonight. Especially conditions being such as they are, we'd love to have you in person, but we really appreciate you making this work for us. And I want to thank all the members. We want to thank all the members who who have kind of worked with us to make our meeting for January work all right. We really appreciate that as well. So I guess if there's no other questions for Garret, we'll wrap things up for tonight. We're hoping to be back to a at least partially live meeting next month, at least a hybrid meeting of sorts. But thanks again for everybody who tuned in and who helped make it a success. We appreciate your dedication to the club, and we look forward to seeing you again next month. So thanks much.
Thanks, everybody.
Yeah.
Thanks for having me.
Thanks, guys. Nice job.